Hello, everybody coming in. We're going to be starting in just a couple of minutes here. Let me remind everybody, this is a webinar format, so you will not be able to see participants. If you have questions during the webinar, go ahead and put them in the Q&A at the bottom or in the chat. And Ranger Lauren will get to those at the end of the talk. And please, everyone, keep your mic muted. So welcome, everybody. I'll go ahead and turn this over to Lauren now.
While you're muted.
All right, thanks everybody. Sorry about that. So thank you so much for being here and for coming to learn a bit more about the Rocky Intertidal program at Cabrillo National Monument. I wanted to share before Anthony gets started with his talk a little bit about where this project came from. And so this project all started with a slide. And it started when I was looking at photos from the 90s of the Rocky Intertidal Zone. And what I saw in those photos was a particular snail. It all started with an observation. And so I was observing this snail in photos and I was thinking, this is like, first, it's a pretty readily identifiable snail, but it's also a snail that we know is been recently introduced to San Diego and was first seen there in the 1950s. And so this project came about by looking at a whole bunch of different photos and trying to see what we could identify over time and to look at changes that were deeper than the percent cover data that we normally record. And I shouldn't say it all started with one photo because it actually started with about 3,500 photos that scientists at Cabrillo National Monument have been taking of the intertidal over time. So since 1990, when this program began, scientists like Keith in the red backpack, who's taking a photo with his iPhone of a plot, have been taking pictures of the Rocky Intertidal plots at the park. And this particular project, which came from observations in these photos over time, also couldn't be done without the help of a lot of different interns and a lot of different people over time. So it's not all just work that we've done recently. And part of this work was done by CNMF intern Rachel Lee, who organized all those 3,500 photos so that we could even start analyzing them for this project. So what I want to end with before Anthony starts is that scientists in parks interns and Cabrillo National Monument or CNMF interns make a huge difference in the science that we're able to do at the park. And I just wanted to express my appreciation for your support of all of these different interns who've done Rocky Intertidal, lizards and snakes, and also fog projects over time. And I'm really excited because Anthony adds on to this legacy of really impactful projects that have been done at the park and that add to our knowledge base. So with that, it's been a really wonderful time working with Anthony during his 12 weeks as a scientist and parks intern. And this presentation is a culmination of all the really cool work that he's done. So with that, I'll turn it over to you, Anthony.
Great, thanks, Lauren. Thanks for the intro. Let me just go ahead and share my screen. All right, just one moment. All right, is that good for everyone? Okay. Cool. So thank you for waiting and welcome to my talk. It's been a really fast past 11, 12 weeks working on this project. In science, that's a really short time frame. So we really just wanted to try and squeeze as much as we could out of that amount of time. And these are basically our results. So what I'm presenting to you today is what we found throughout this project. And before I begin, I just want to ask this really simple question of, should I stay or should I go in regards to range shifting species that we have been encountering in our tide pools here at Cabrillo National Monument? So to start things off, just wanted to introduce myself very quickly. I finished my master's thesis at San Diego State University this past summer. And before that, I've always had a very strong passion for marine science, whether it's through research or through recreation. Um, Living by the coast has been a very impactful part of my life. And hopefully it's not just me, right? Because you all hopefully have experienced some form of use from the ocean, whether that's through food or transportation via shipping or going there on vacation. The ocean plays a very large part in all of our lives, which is why it's pertinent that we manage and preserve these resources. Obviously, however, we don't always do the best job at that. And a wide variety of threats are currently pressuring our natural resources. So I can go into things like overfishing and pollution. But for today's talk, I'm going to be looking more towards the future and what that holds in regards to climate change, and as a result of climate change, range shifting species. But before I get too far into that, what I have found is that when we see a problem, the best solution to tackling that problem is to learn about it. And so that's what I did. Here is me and several other very intelligent scientists conducting some research in the intertidal. This picture here is us at a 3 p.m. low tide, which is all groovy. And this picture is of me at a 3 a.m. low tide. So a bit of sacrifices were made in terms of sleep. But overall, what I was interested in was this macroalgae right here, Sylvatia compressa, also known as the golden rockweed. It's found in our intertidal communities here at Cabrillo. And what I wanted to learn was, what impact would climate change have on this species? So if I can just get this video to play. So just to ground everyone, here is Sylvatia growing in our tide pools on top of these boulders. And just want you to pay attention to the fact that where it is not growing, the rock is bare and it's covered in barnacles. However, if we take a closer look at Sylvatia and we pull some of it aside and we just look at how the understory community looks where it's present, you'll note that it is harboring a very diverse understory. And that's due to the fact that it's trapping moisture and keeping the substrate in a condition that's conducive for allowing these organisms to grow when they're exposed during the low tide. So obviously, Silvicia is hosting a very important community. However, through my research on climate change and specifically on climate change on Silvicia, we found that Silvicia is negatively affected by climate change. And that, as a result, is due to the fact that temperature, sea surface temperatures in particular, are increasing steadily with climate change. So with that comes physiological stress to organisms that are experiencing these rising temperatures. So Silvicia is one of those afflicted. Perhaps a more readily recognizable example of the effects of climate change is on sea stars and how climate change caused the sea star wasting disease that we observed back in the mid 2000s or 2010s. And so now we're kind of looking at this issue, right? As climate change worsens, how will our natural resources be affected? How can we manage and mitigate the loss that we might expect going into the future? One result of increasing temperatures as a result of climate change is that species are going to have a couple of options that they have to choose from in order to deal with these rising temperatures. The first and probably not very preferable option is they can die. If you think about species living within a very narrow range, if it gets too cold or if it gets too hot, they get unhappy, their metabolisms don't work as well, and they just die. Two is they can adapt to the changing temperatures. They just learn to deal with it in one way or another. And their third option is to pack everything up and leave. All right, so whichever option allows the most species to not go extinct would probably be the one that is the best option for everyone involved. And today I'm going to look at what happens when species move around and change from one community that is no longer inhabitable for it and moving to a new community where the temperature is not as extreme for that species. So if we think about species packing everything up and leaving, it does call into question exactly what effect they're going to have on these new communities. And when you have one species leaving its native home range and shifting into an entirely new area, that's very reminiscent of the patterns that we've observed with invasive species. And so invasive species are organisms that, or sorry, species that come from a different place and enter a new area where they are unchecked, right? There's either a really large food source that they can take advantage of, or they lack a natural predator in this new place and they're just able to multiply and reproduce and essentially take over and cause negative effects on the ecosystem that they've entered. Generally, we think of invasive species as ones that we as humans transport. So things like hitchhiking in the barge of cargo ships or being brought over from a different country to be planted because we think it looks pretty, and then has all of these negative effects. The distinction between range shifting species and invasive species is that these species are shifting on their own, not necessarily as a result of something that we have done. But because they are entering a new space, there's going to be a whole suite of different interactions that we have not predicted, and understanding exactly what effect that will be is going to be important if we want to know what their impact will be as climate change worsens and as more species shift their range in response to increasing temperatures. So one example to hopefully drill in what I've just talked about is mangrove forests. Mangrove forests are a tropical species. They form these aerial roots that jut into the the intertidal area. And these roots provide a lot of structure and habitat complexity that allows juvenile fish to use as a nursery ground. And so these mangrove trees and the ecosystems that they create are really critical for maintaining that biodiversity in these areas. However, as temperature has increased, mangrove trees have been shrinking in their range, and alongside deforestation and development of coastal areas, we've seen a really large decline of mangrove trees. However, we have also noted that mangrove tree populations have been shifting northwards towards the poles, where temperature isn't as extreme, away from the equator. This is a good thing because it's preserving that critical habitat structure that they're providing. However, there are unexpected consequences as a result. And one example of one such consequence is that the mangrove trees are facilitating these lionfish down here, which are an example of an invasive species that has been found in the Caribbean. The lionfish are very voracious predators. They love to prey on juvenile fish, and they've caused a lot of fisheries in the tropics in that area to be destabilized by their presence. So while the mangrove tree expansion is overall a good thing, it's also facilitating these new interactions that we didn't intend for by allowing this invasive species to continue spreading. So what do we do when we don't know what's going to happen, essentially? I can't speak for everyone, but here at Cabrillo National Monument, there are several methods that we have employed to handle invasive or range-shifting species. On the more hands-on side of things, we have the weed warriors here at Cabrillo National Monument. These weed warriors go out and pull out invasive species and replant native species as a way to preserve our biodiversity here at the park. And so that's going to be more of a direct approach to managing an already invasive species. We can also monitor incoming species and assess their changes so that we can then employ management efforts such as weed warrior removal and revegetation. So in the intertidal area of Cabrillo National Monument, we have marine monitoring. Marine just stands for the Multi-Agency Rocky Intertidal Network. It's a long-term data set that Lauren alluded to, spanning back several decades, encompassing over 3,500 photos. And what marine monitoring is just looking at the change in our tide pools over a very long period of time so that we have many comparisons that we can make and so that we can observe changes. So I just want to briefly introduce the methodology of marine monitoring. Essentially, we have long-term plots that are marked by bolts. We go out there and we revisit those plots every year, lay down this transect, and then we count and score what we find in each plot to see the change over time. And this is just a way to measure what the dominant makeup of each plot is, what its composition is, and just seeing how it's doing. So this gives us a really unique opportunity because now we can observe changes throughout time. This is the same plot in 1995 here on the left. And as we move to the right, you can see that these barnacles this cluster of barnacles is growing and growing and growing. And so we can see species growing, we can see them decline, and we can see them come and enter when previously they weren't there before. So one such species that has shifted its range into Cabrillo is Mexicanthina lugubris. And that's going to be the topic of my project and of the rest of this talk. Mexicanthina is also known as the dark unicorn whelk. You can maybe see it here, but it's so called because of this little prong that juts out of its shell that it can use to pry and get leverage on other intertidal organisms that are stuck really tightly to the rocks. And it's doing that because, unlike most snails, these guys are carnivorous and do enjoy a little meaty morsel from time to time. And what we've observed is that originally they are native to Baja California, Mexico. However, as the temperature has warmed, we believe that that has been correlated with their observed shift by the mid 2000s past Laguna Beach in California. So now they're in Southern California. And we've observed them at Cabrillo via the marine photos and by people going down and observing and picking up shells and being like, hey, this wasn't here before. When Mexican thyina occurs in the communities that it is found in, it interacts with a whole assortment of intertidal species. So just for example, Mexican thyina, because it is a carnivorous snail, actually preys on bivalves and gastropods crustaceans, such as mussels and barnacles, pictured here. In addition, we have native whelks as well, which have always been here, and they prey on a similar suite of organisms. So when you have two species competing for the same food source, they're known as competitors. And so Mexican thyina is now an introduced competitor for our native whelks. And beyond that, Mexican thyina can serve as prey for a number of higher trophic level predators. And beyond that, when they die, their shells form shell sediments. If their shell remains intact, they can be used as hermit crabs or used by hermit crabs. And so what that means is that this species has entered a new area and it is interacting in a variety of different ways with the native organisms found there. And what we want to know is exactly what is it going to be doing as a result of its presence or its absence. So we figured it might be wise to monitor Mexican thyina. When Lawrence saw the whelks in the marine photo plots and realized that we could go through those photo plots and gather information about the snails inadvertently because it was just noticed to be in those plots coincidentally, that we could draw some information and try to figure out exactly what they're doing. So this is a very opportunistic study. Essentially, what I did was I went back through these photos to look for the presence of these whelks. And then I wanted to pair that with a variety of factors that would best correlate and provide information about these whelks themselves, just based off of the pictures alone. So what I came up with was one time. I believe that the Mexican thyina population is changing over time, and that the population is changing as a result of temperature-induced. The range shift has been induced by a changing temperature. So the second question is whether or not Mexican thyina abundance is correlated at all with sea surface temperatures. The third factor I wanted to look at is the location in which Mexican thyina appears in, simply because wherever these whelks are appearing is where they'll likely have the most impact. And lastly, within the location or habitats that they preferably or preferentially choose, which species found in that area will Mexican thyina associate the most with? So these were the four sort of factors that I wanted to pair all of the Mexican thyina sightings alongside to try to draw some preliminary conclusions. So here's just an example of one of the photo plots that I poured over. What I did was I overlaid a grid, and this allowed me to standardize my search time just by going through each panel one by one and trying to locate any whelks. So if you want to give your hand at looking for a whelk, I'll give you a couple seconds. And if you're able to find it, that's great. I really could have used your help because there are a lot of photos to go through, but no worries because it got done. But hopefully you're able to see it. He's right there. And so I would continue this process for all of the photo plots that we've taken over the past several decades. Some plots were a lot easier than others. So for example, that last plot had 1. This one had about 20 to 21 by my best count. And this is actually the plot with the most amount of whelks found throughout those years. So this is an example of like a max whelk density. So all that said, all that data was compiled and then paired with these factors. And so here are what the results look like. So for the first question, is the population changing over time? Yes. So what I have here on the x-axis is the year, and on the y-axis is the density of Mexican thyna per meter squared. So that's the size of each plot. And if we look at it over time, we see a steady increase from the 90s up until about 2005, 2006, where the population peaked before a steady decline over the years. So this is just a look overall at what the population looks like throughout time. So now we would like to know if any of that pattern is at all correlated with temperature. So what I have here is I've generated the average sea surface temperature collected from buoys off of Scripps Pier over time. And as we can see, there has been a steady increase in the average sea surface temperature. However, as you might remember from the previous graph, that the Mexican thyna change over time wasn't a very linear one. So when we plot it against temperature here in the x-axis and density on the y-axis, we don't really see much of a correlation. So that does go a bit against our initial hypotheses. But I'll talk more on that in just a moment. For my third question regarding which habitat is preferred by Mexican thyna, I would just wanted to ground everyone exactly with how the plots themselves were distributed. So Cabrillo National Monument, the intertidal area is split into 3 distinct zones. Zone 1 is right off of the public trail, and that's where the most amount of visitation occurs. Zone 2 is still open to the public, but fewer people venture over to that zone, so there is much less visitation. And lastly, Zone 3 is entirely closed off to the public. So because the plots are arranged in this zonation, we can try to pair that with the Mexican thyna data. And if we see the fewest amount of Mexican thyna in Zone 1, that might give some indication that they are affected in some way by human presence. Zones 1 and zones 3 are most structurally similar in terms of geography and geology, sorry. And that's going to be distinct from zone 2, which is much more comprised of cliff faces and boulders than zones 1 and 3. So if zone 2 shows any distinction between zones 1 and 3 in terms of Mexican thyna abundance, then there might be some habitat type preference coming into play. Secondly, when we are monitoring these plots, there are 4 plot types, and these are to monitor target species, and these were all set up back in the 90s. That's when all of these decisions were made. So within each of the 3 zones, there are 4 plot types. The first plot type is labeled as Balanus/Thamylus, aka acorn barnacles. That's the species being monitored in those plots. We also have Middleus or mussel plots, Polycipes or goose barnacle plots, and Sylvatia or golden rockweed plots. And each of these 4 plots have 5 to 6 replicates for a total of 60 or so plots that we monitor yearly. And each of these plots is set at a different elevation or tidal height. And so what that means is I can pair zone data with Mexican fauna distribution, plot type with Mexican fauna distribution, and elevation with Mexican fauna distribution to kind of get at which habitat type is most preferred. So for the first part, we found that zone 2 had the greatest density of Mexican fauna, whereas zones 1 and 3 were a bit more comparable to one another than to zone 2. So what that tells me is that human presence doesn't seem to have too big, or it doesn't have too big of an effect on Mexican fauna, because if that were the case, then zone 3 would probably be closer to zone 2 or even higher because of how few humans actually make it out to that area of the intertidal. And it also tells me that zone 2, because it's structurally distinct from zones 1 and 3, might play some form of preference for Mexican fauna. After that, I want to look at the plot type. So here on the y-axis are the 4 different plot types. So Thamylus, Middleus, Polycipes, and Sylvatia, plotted against the density of Mexican fauna. And we find another distinction in that Polycipes plots had a higher average density of Mexican fauna than the 3 other plots. And if we look at elevation, we can see that there is a bit of a linear relationship between the tide height and the abundance of Mexican fauna. So when we think about elevation, because the substrate slopes downwards as you move towards the ocean and upwards as you move away from the ocean upward towards the cliffs, we are seeing that Mexican fauna seems to prefer those upper elevations a bit further away from where the water level is. And if we look at the specific plot makeup, we do note that the Polycipes plots are all of a higher elevation than the other 3 plot types. So there may be some teasing apart that needs to be done about whether or not it's the elevation that the Mexican fauna are after, or if they're after something found within the Polycipes plots, such as the Polycipes themselves, which could serve as a food source for these carnivorous whelks. And then we did have a bit of an outlier. That's zone 2 Polycipes plot 5. That coincidentally is the plot that did have that I showed previously with those 20 plus whelks in them. So that answers some of our questions about location. And then for my last question, I wanted to ask, which species does Mexican fauna associate the most with? So to do that, I took the percent cover data that was previously determined for each plot. And the way percent cover is scored is this grid is placed over the plots in the intertidal, and we have a person scoring. So wherever the lines intersect, whatever is beneath that point is recorded, that species is recorded. And from there, we can calculate the percent cover, what percent of the plot was covered by each species. Because the target plots didn't always have the highest percent cover of the target species, right? So this is a Polycipes plot, and Polycipes is shown here. It's taking up about maybe 10% of the total plot area. I wanted to know if Mexican Thyna was associating more closely with some species more than others. And so because the percent cover was not evenly distributed for every species, and because Mexican Thyna can move around and decide to aggregate and associate more closely with other species, I also, when I was scoring the Mexican Thyna, I recorded the closest neighbor or whatever it was in contact with. So, for example, this one up here is in contact with Polycipes, so I would mark it down as a Polycipes Mexican Thyna. And this was just a way for me to see whether or not there was any sort of preference for what kind of species Mexican Thyna would associate with. And so for this next plot, given both of those protocols, we can try from there to see whether or not the plot composition lines up with the Mexican Thyna use. And if there's any disparity between those two, then we could say that Mexican Thyna is either using one species or is more frequently observed in contact with one species more than another. So for example, I have here on the y-axis of this graph the proportion of the plot that each species is taking up. And paired with that chart is the proportion of the species that Mexican Thyna is in contact with. So for Thamylus, if I wanted to look at whether or not Mexican Thyna is aligning itself with barnacles more often than not, then I would see if the corresponding proportions of barnacles is similar. So in the Thamylus plots, the barnacles that are the target species of those plots are taking up this level of proportion relative to the plot as a whole. And when we look at Mexican Thyna usage, we see that it's pretty similar. So it seems like it's distributed pretty evenly, and that Mexican Thyna isn't associating too strongly with Thamylus. However, if we look at the Middleless plots, in the Middleless plots, mussels or Middleless don't take up too much of the total amount of space in those plots. They're observed a little more infrequently, or they don't cover up as much space as other species might. However, when we found Mexican Thyna in the Middleless plots, what they were most in contact with relative to the rest of the plot is Middleless. So from here, we can say that, like, even though Middleless was found in a lower proportion in these plots, the Mexican Thyna contact was much higher. So we can then think about, well, why exactly are they in contact with Middleless so often? Are they using it for cover? Are they using it as a food source? What's going on? Similarly, with Middleless, we found a similar pattern with the Polycipes and the Polycipes plots. The goose barnacles didn't take up too much space relative to just bare substrate. However, the proportion of which Mexican Thyna was in contact with Polycipes was much higher. And so we can think about how Mexican Thyna is using Polycipes in a similar manner as it is using Middleless. And lastly, for Sylvicia, whenever we found a Mexican Thyna in a Sylvicia plot, it was more often than not found amongst Sylvicia fronds. So almost 100, more like 75% Sylvicia usage by Mexican Thyna, even though it was closer to a 60% total proportion in terms of Sylvicia cover. All right, so that kind of gets at what we want to know about Mexican fauna and how it's using each plot type, and within each plot type, which species it is associating most closely with. And from there, we can gather all of the data that we got and all of the data interpretation from those graphs and ask, well, what did we find out about Mexican fauna? What have we learned? And so with that first plot, we did note that over time, the population, at least here in Cabrillo, appears to be declining. Whether or not it's declining because its population overall is going down, or because they are continuing to shift their range over time, is currently unknown. It's entirely possible that they are moving out of Cabrillo and even more northward up into more southern central California as the weather gets warmer. They could be shifting back down south towards Mexico. We're not quite sure. But over time, it appears that Cabrillo is more of a rest stop for this population if that decline continues. Contrary to our hypothesis, temperature was not a powerful predictor for the abundance of Mexican fauna. If it were a linear relationship, we would have expected Mexican fauna to be increasing in abundance and density over time because temperature has been increasing steadily. We did not find much of a correlation, and so as of right now, we should probably look at other hypotheses as to why Mexican fauna is shifting its range, if not temperature, then what? As far as the location of Mexican fauna, we found the most amount in Polycipes plots that were found in Zone 2. And whether or not that's because of elevation or because it is there to prey on these goose barnacles remains unexperimented and unstudied. And then lastly, the biota that were most associated with Mexican fauna were in fact the mussels in the Middleist plots and Polycipes in the Polycipes plots. Right, so that's kind of just like an overall summary of my results. However, long-term monitoring is only part of the bigger picture. Because this was an opportunistic study, we just saw these whelks hanging out in our plots and just decided to go back and look at them. That means that none of the plots were set up and designed specifically to monitor Mexican fauna. So one such shortcoming is in those Sylvetia plots. When you pull back Sylvetia and you look at that very diverse understory that it hosts, Mexican fauna is part of that community. However, in the plots themselves, because we were just looking at pictures and not moving aside the canopy or anything like that, it's very likely that we've underestimated just how many Mexican fauna are found in those Sylvetia plots. So it's totally possible that Mexican fauna is more associated with Sylvetia than Polycipes plots, but currently that remains something that will need to be studied moving forward. So that's just one example of a shortcoming. And then other examples are really experimentally going in and finding out exactly how it's affecting the surrounding native tide pool organisms. Simply because I'm looking at photos, I can only make hypotheses as to whether or not the Mexican fauna are eating whatever they're touching, or if they're just harboring there and waiting out the low tide because there's more moisture associated with some species more than others. Obviously, if they were preying on other species, that would be a much larger impact than if they were just using a species as shelter, right? So really answering the question of whether or not the Mexican fauna are having a negative impact or not would require some deeper dives. So just to round us out, should Mexican fauna stay or should they go? before I can really answer that question, we should continue monitoring the Mexican Thina population, but with a more focused intent, right? Setting up surveys that will really capture exactly how the population of Mexican Thina is changing, whether that is by setting up surveys designed to count Mexican Thina or some other method. That would really allow us to get at that, because in the pictures, we're not targeting Mexican Thina at all. So there could be pockets and large populations of Mexican Thina that exist outside of those photos. We just never are able to catch that. Pairing the long-term data set with experimental studies to really figure out how they're competing with other species, which species they are serving as prey for, which species they are preying on, and looking at those interactions in particular will provide us with the best insight as we pair that with data about how the population is changing, right? So how much or how many Mexican Thina are here in one place and what effect they are having exactly. Lastly, Mexican Thina have very established roles in the intertidal communities here at Cabrillo. Like I mentioned, they've been here for at least the past couple decades and likely even longer. The roles that they play have already been established, whether that be prey, predator, or competitor, but these interactions could change in the future as climate change increases temperature, as sea level rise increases, as new species shift their range or become invasive in Cabrillo. How is Mexican Thina going to interact with all of those changes? And could it be beneficial or harmful? That remains to be seen. So while maybe an unsatisfactory answer, it really is that it depends and that we really need more information on these little guys. As far as whether or not they should stay or should they go, my best foot forward is that we don't pull them out and do anything of that sort of nature, but we continue to monitoring them and see what happens from there. But all that being said, I would like to make some acknowledgements to the Cabrillo National Monument Foundation, National Park Service, and Conservation Legacy for giving me this really cool opportunity to be out in the intertidal, get paid for it, and helm a really cool project. And I would also like to thank the resources management team for sharing some office space with me. It's been a really fun time being there, running around Cabrillo, down to the tide pools, back to the office, down to the tide pools, just making it a really welcoming environment to work and conduct science in. So with that, I'll take any and all questions. I don't think.
Excellent. I already have a question. Okay, so for those participating in the webinar portion, I can take questions either in the chat or the question and answer box, and I'll read those to Anthony, and then we'll have answers. So the first question is from Patricia, and she asks, are the snails monitored at all in their original location? And if so, has the population declined there?
That's a great question. As far as local monitoring in Baja, I don't know if, I'm sure there have been studies on the whelks. Marine monitoring in Baja has only like recently extended down there. So if their protocols are anything like ours, then Mexican Thina currently are not on that list of monitored species. And that's kind of what the purpose of this project was to ask whether or not to include monitoring of Mexican Thina in marine monitoring. So moving forward, it's possible, if I was persuasive and argumentative enough, that Mexican Thina can be added to the monitoring list. But at present, While I can't speak for Mexican science, marine monitoring, at least in Baja, currently is not looking at Mexican fauna. And then as far as whether or not they've decreased in their native range, I believe the answer to that is yes. Their southernmost range has been depleted of Mexican fauna, while their northern range continues to expand up into the Californian coast.
Thanks. Let's see, I'll give everybody a minute to type if you have more questions. Okay. The next question is, did you notice a decline in species that are associated with Mexican fauna?
So the species associated that we pulled from that data were mussels and goose barnacles.
Mhm.
And I believe the data for the percent cover of mussels has declined over time. However, it's really unlikely that that was caused by Mexican fauna. I believe it's most likely a result of visitation or higher visitation causing mussel populations to decline. That coupled with the fact that the mussel plots are found at lower elevations where Mexican fauna is less likely to be found means that they are probably not actively targeting mussels, at least where they're found in their highest numbers. Right, so as far as mussels goes, I don't believe so. And then for the goose barnacles, the polycipes, I haven't really noticed much of a decline in polycipes. If anything, they've been doing pretty well. So whatever they're eating, they're not eating to extinction, like depleting, as of right now, time being.
Thanks. The next question is, has the snail continued to move north, and has it been discovered in other places?
Yes, it has been continuing a northward trajectory. There's been research done, I believe is Piper Wallingford's paper, looking at sites north of Cabrillo, where the whelks have been reported at pretty low densities in the mid-2000s, but are now increasing in abundance on subsequent visits. So the range shift itself does appear to be, tracked. It appears to be tracking and trending northward, while the southern range appears to be declining. So we are seeing kind of like this movement northward of the Mexican fauna population.
Yeah, and if I can add a little bit to that, I think you showed an iNaturalist graph earlier, that showed where there were reported sightings of Mexican fauna. While the northern edge of their range right now in the scientific literature, which often lags behind observations, right? Because it takes a really long time to go from observation to published paper, is I think 1,000 Steps Beach and Laguna Beach. There are iNaturalist observations that go like all the way up to the Los Angeles area. So I wonder, and I think that's something that we'll have to do a little bit more investigating, right? I wonder how far north they have expanded and how fast they're shifting, which is pretty fast from, I think they went from San Diego, which is where they've been since like the 50s, to Orange County by the 2000s. And for a snail, it's not very slow.
Yeah, especially if you take into consideration that they lay their eggs locally, which means that they don't really have a larval phase to get swept northward. Means that like they are opportunistically moving northward. The ones that do make it northward survive to reproduce more, but it's a much slower process than if they had pelagic larval gametes.
Yeah, which means that the larvae float around in the ocean for a period and develop before they land and start living in the intertidal. Cool. Well, that is all the open questions. And thank you so much, Anthony, again, for giving this presentation and also for working with us. It's been really awesome. And I'm excited that we still get to write a paper next
Yeah,
week.
it'll be fun. A great time for everyone.
Yeah, and with that, I'll hand it back to Dan with some announcements about future events.
All right, thank you. Just a quick update that the next talk, Natural Speaking Talk, will be on Thursday, January 19th. Hopefully it will be another hybrid meeting. So that is not available to RSVP yet on the foundation site, but probably by next week it should be out there. And then I also would like to just say thank you to the foundation and Anthony. That was a very interesting talk. So thank you very much. And thank you everyone for attending tonight.
Yes, thank you for coming to listen to me talk about some snails. It was a great time. Yeah.